You are listening to Professor Megan Workman. Welcome to this lesson on membrane transport, which refers to the movement of molecules across a plasma membrane. In this lesson, I will describe membrane structures that facilitate transport. These include integral membrane proteins called protein channels and carrier proteins. Then I will explain different mechanisms of membrane transport, distinguishing between passive and active types. And finally, we will apply this new knowledge about membrane transport to understand how SARS-CoV-2 virus enters host cells. Proteins are the membrane structures that both facilitate and regulate transport of molecules from one side of a plasma membrane to the other. The function of these proteins is to allow the cell or the membrane-bound organelle like a mitochondria or a lysosome or the nucleus to control the composition of its interior space. In other words, it allows a cell to maintain homeostasis, or the chemistry necessary to support its functions. So this might include maintaining proper pH balance, maintaining concentration of solutes within the cytoplasm, including gases like oxygen, carbon dioxide, including fuel sources like glucose, and importantly, also including chemical byproducts that might poison the cell, like waste products. Next, I'd like to illustrate this with a couple of examples. Recall from a previous lesson that integral membrane proteins are those that are permanently embedded within the plasma membrane. That's what you see here. You see an integral membrane protein. This one happens to be involved with transport, and it's embedded within the purple phospholipid bilayer. Notice also that this protein is comprised of two chemically distinct parts. If the protein is a hallway, you can imagine that the wallpaper decorating the hallway is made of polar parts, through which polar molecules can easily travel, while the underlying scaffolding of the hallway is comprised of nonpolar parts, and these are the parts of the protein that are interacting with the hydrophobic core of the plasma membrane in which they are embedded. This is a convenient situation for these cartoon water molecules because they can zip right through the polar internal space of this transport protein. So here you're seeing water diffuse through a protein channel. Diffusion through a protein channel is called facilitated diffusion. To be clear, this transport protein is facilitating or helping these polar water molecules diffuse from one side of the plasma membrane to the other rather than getting blocked by the hydrophobic core of the plasma membrane. Some example molecules that require facilitated diffusion include polar molecules like water, charged molecules like ions or amino acids, and really big molecules, large molecules like carbohydrates that otherwise wouldn't fit. They just wouldn't readily diffuse across the plasma membrane, so they have to find a wide hallway to pass through. It's important to note here that small enough polar molecules, in fact including water, can diffuse across the plasma membrane, but because of that hydrophobic layer, they diffuse rather slowly. So slowly, in fact, that it would not allow the cell to maintain homeostasis. So the integration of transport proteins into the plasma membrane allows a cell to control the rate at which molecules pass either into or out of the cell. An example of a transport protein that performs facilitated diffusion in this way is called an aquaporin. which is a protein channel. Aquaporins are open at both ends, meaning they allow the bidirectional movement of water both into or out of a cell depending on the cell's hydration status at that moment. So aquaporins allow the cell to maintain osmotic balance. Imagine a situation in which the water is moving in the opposite direction. In this example, water is being transported from where water is in low concentration to where water is in higher concentration. So here the cell is purposefully importing, in this case, water. There are a number of things a cell might want to purposefully import, like glucose, sequestered out of your bloodstream to support the metabolic processes inside the cell. So even when you're hungry and your blood glucose level is low, your cells are still spending ATP energy to import glucose from where it's in lower concentration outside of your cell to where it's in relatively higher concentration inside of your cell. So in this example, the molecule is imported against the concentration gradient, and therefore that requires energy. This is called active transport. Active meaning requires energy. You could contrast that with passive transport, which doesn't require energy. Examples of molecules that are selected for active transport are things that are useful to the cell that it's actively importing, or waste, perhaps poisonous waste products that are being actively exported. So food is often actively transported in, like glucose. Ions, like electrolytes, are often actively transported. Toxic waste is often actively exported. A real-life example of an integral membrane protein that serves this function is called GLUT4. It is a carrier protein that actively transports glucose from your bloodstream into your skeletal muscle cells or into your adipocytes. Here is a cartoon to give you examples of things that either absolutely require facilitated transport, meaning a protein hallway. Big polar molecules like glucose ain't gonna diffuse across the plasma membrane without the aid of a transport protein. Similarly, charged molecules like ions will be repelled by that hydrophobic core of the plasma membrane, so they absolutely require the help of a transport protein. Smaller molecules, like gases or hydrophobic molecules or teeny tiny polar molecules, can diffuse through on their own. But in the case of especially polar molecules, it happens so slowly because they become blocked in the hydrophobic core of that membrane that they are aided. Diffusion can happen more quickly across the membrane if it's facilitated through a protein hallway, through a transport protein. So facilitated transport, which is transport through a protein, speeds up the process for these littler molecules. Now I've thrown a couple of terms around: protein channel, carrier protein. So I'd like to take a moment to distinguish between these two concepts. Both are types of transport proteins. A protein channel is relatively less selective, meaning lots of protein channels allow multiple types of molecules to pass through. Protein channels are only involved in passive transport, meaning they allow molecules to move along the concentration gradient only. Some are unidirectional, but some can be bidirectional, like the aquaporins I mentioned earlier. Bidirectional means that the protein channel allows the movement of molecules both into and out of the cell. Contrast that with carrier proteins, which are selective. Carrier proteins transport only one specific type of molecule, and this is accomplished by having a binding site on this protein itself to which the molecule that is hoping to pass through the hallway is chemically affiliated, and when it's bound, it changes the three-dimensional conformation of the carrier protein itself in a way that releases the molecule to the other side of the membrane. Carrier proteins can be involved with either passive or active transport, and they're always unidirectional. So they're selective and they're unidirectional. The example carrier protein I gave previously is a GLUT4, which actively transports glucose into certain cells in your body, including skeletal muscle cells. To review, proteins facilitate transport of molecules across plasma membranes. This is accomplished by creating a sort of what I'm calling a hallway through which molecules can pass. Sometimes the walls of the hallway are hydrophilic, which allows polar molecules to pass more easily across the plasma membrane, because remember, they would normally get held up in that thick hydrophobic core of the plasma membrane. The hallway could be really wide, allowing large molecules to pass through. Or the hallway might be gated. Perhaps a key is needed to open the hallway. A type of key that could open a protein hallway, a transport protein, is called a ligand. So perhaps the transport protein is functional only when this ligand attaches. That's what you see in the image here. When this ligand chemically binds to the transport protein, you can see that the hallway opens up, allowing the passage of molecules. Perhaps the hallway only opens when mechanically forced to, when pressure is applied to the membrane, or perhaps it opens when a certain electrical potential occurs across the membrane. So far, we've discussed high precision transport mechanisms, including facilitated diffusion and active transport, during which a low number of molecules pass across the membrane at a controlled pace, or in the case of carrier proteins, just one molecule passes at a time. Here are some images reviewing how this high precision type of transport works. In the left panel, we see a number of different types of passive transport, during which no energy is required because molecules are moving down their concentration gradient. Whereas on the right side of the screen, you see active transport, which is any movement of molecules against their concentration gradient, and this always requires cellular energy, which we call ATP. Facilitated diffusion happens through different types of transport proteins. The first one here you see in the middle is called a protein channel. You can see the molecules passing through this protein channel. Next to it is a carrier protein. You can see its binding site that allows a specific type of molecule to pass one at a time unidirectionally. And then, do you know what type of transport protein performs active transport? Yes, you do. It's a carrier protein. The last thing I want to highlight on this image are all of the things over here that are simply diffusing across the plasma membrane without the aid of a transport protein. This is called simple diffusion. Gases like oxygen and carbon dioxide can diffuse across the plasma membrane without the help of a protein. Hydrophobic molecules, especially small ones, they can easily diffuse across that hydrophobic core of the plasma membrane. And tiny polar molecules too, but remember that the pace of movement of polar molecules is slowed by that hydrophobic core of the membrane. This brings us to a lower precision type of membrane transport mechanism, called endo, or oppositely exocytosis. Endocytosis and exocytosis refer to the bulk movement of molecules either into or out of the cell, respectively. Here is what exocytosis looks like. Imagine a situation in which a cell perhaps produces a cellular product. Maybe this is a gland that is producing hormones for export to the rest of the body. So in this example, the green blobs might be hormones. Your cellular machinery produces the hormones and then packages them into a vesicle, which is surrounded by its own plasma membrane, which is chemically identical to the plasma membrane of your cell membrane. So the vesicle floats over to the cell membrane, fuses with it, and then releases its contents into the extracellular space. So this is bulk movement of these hormones out of the cell. In addition to hormones, large molecules that the cell wants to export could be gotten rid of in this way. And interestingly, this is also one way that viruses are shed. If this cell were infected with a virus and it had turned your cellular machinery against you in order to be a viral producing factory, it could envelop all of these viruses in a vesicle. The vesicle flows to the cell membrane and they're all released into your bloodstream over and over again by multiple vesicles. Gross. Next, let's consider endocytosis, which is the bulk movement of molecules into the cell. During endocytosis, the plasma membrane curves in on itself, creating a small divot in which a number of molecules fall. Then the cell membrane eventually pinches off on itself and it takes in a vesicle. So these green blobs are now representing things that the cell might normally want to take in bulk, like food. But another thing that the cell might accidentally take in during this low precision type of importation of molecules, low precision molecular transport, are viruses. So this is one way in which non-enveloped viruses sneak into the cell. Here you see one hiding among perhaps food molecules, and this is one way it gains entry into host cells. So now you've learned 4 mechanisms of membrane transport, including facilitated diffusion, active transport, endocytosis, and exocytosis. Next, let's turn our attention to the ways in which cells can regulate all of these transport mechanisms in order to exert even more control over the types of molecules that they are importing or exporting, and also the pace at which these molecules are passed across the plasma membrane. You may not be surprised to learn that proteins are involved in the regulation of all of these transport mechanisms. And I say it with that inflection in my voice because it is proteins that are the workhorses of the cell. They have many different functions. You've already learned that one function of integral proteins is transport. These transport integral proteins perform facilitated diffusion and active transport. Another type of integral protein is a receptor. These receptor proteins have the function of inducing endo or exocytosis, and in this way regulating when the cell performs bulk import and export of molecules. Additionally, you previously learned about peripheral proteins, and I want to revisit them here because peripheral proteins play a role in the regulation of whatever integral proteins they might bind to. So here in the cartoon I'm borrowing from the Amoeba Sisters, there's a little peripheral protein wearing a party hat bound to a larger transmembrane integral protein. This little peripheral protein could act like an on-off switch for the function that the integral protein normally performs. So perhaps when this peripheral protein is bound, it's shutting down the facilitated diffusion and thereby controlling what the passage of that molecule that would be normally either going into or out of, perhaps both, across the cell membrane. So peripheral proteins regulate the functionality of protein channels, carrier proteins, and also receptors. So if a peripheral protein is bound to a receptor, that prevents the receptor from inducing endo or exocytosis. Next, let's apply what you've learned to a real-life example. This image demonstrates the two ways that SARS-CoV-2 virus, which is the virus that causes COVID-19 disease, can enter host cells. I want to use this opportunity to review several key terms that you learned in this and previous lessons. So it's going to be jargon heavy. Let's first evaluate the left side of the screen, in which you can see the coronavirus entering the host cell via endocytosis. How this works is that glycoprotein spikes on the viral envelope bind to a receptor on the host cell. And once bound, this receptor induces endocytosis. We can add even more jargon to this explanation. Because glycoprotein spikes are both integral proteins that have glycans attached. They're glycosylated. This is what makes them a glycoprotein. So these glycans are signaling information that is received by the ACE2 receptor, which is itself an integral membrane protein, but on the host cell. The ACE2 receptor is there because it normally binds to something else that the cell healthily imports. So the SARS-CoV-2 virus evolved to mimic the glycan that is normally found in your body and thereby co-opt the normal function of the ACE2 receptor. So once this glycan is bound to the ACE2 receptor, you can see what happens. The cell membrane created a little divot. Normally it would be around healthy molecules that the cell needs to import, but here it's around the coronavirus. And it takes this virus in as a vesicle, which later wreaks havoc on the cell. Let's contrast this endocytosis entry mechanism with membrane fusion on the right. During membrane fusion, glycoprotein spikes on the viral envelope bind to receptors on the host cell, causing the envelope to fuse with the cell membrane. This is made possible because the viral envelope is actually made of plasma membrane, chemically very similar to the plasma membrane that comprises your cell membranes. Once bound, then your cell actually grows a little bit by the diameter of the virus and now contains viral glycoprotein spikes. This is great news because the immune system will come along, recognize these glycoprotein spikes as being of viral origin and attack the cell, shutting down the infection. And here I thought it would be interesting to show an electron micrograph. So this is a true life image provided by the National Institute of Allergy and Infectious Disease that has electron microscopes. And they actually have a Flickr account, so you can go to this URL I've included at the bottom of your slide and see all sorts of amazing images like this one. But I chose this one to show you because On the right is the coronavirus. You can see all of its glycoprotein spikes, and you can also clearly see the two that are presently docking with this soon-to-be infected host cell. You learned a lot in this lesson. You learned that transport is both facilitated and regulated by proteins. Specifically, you learned about protein channels, carrier proteins, and peripheral proteins, which sometimes serve as regulators of channels and carriers. You also learned that whether energy is required for transport depends on which way the molecules are moving in relation to their concentration gradient. Specifically, you learned that active transport uses energy to import or export molecules against their concentration gradient, while passive transport refers to transport of molecules simply down their concentration gradient, so no energy is needed. You learned about the bulk movement of molecules into or out of cells called endocytosis and exocytosis. And you learned the two mechanisms by which SARS-CoV-2 virus can enter host cells. These are endocytosis or membrane fusion. You also got to see a cool real-life image of SARS-CoV-2 about to infect a host cell.